Can everyone hear me okay? So thanks very much for the introduction, Ingrid. That's used up most of my time. And thanks for the acknowledgement to the traditional owners, the people of the Kulin Nations. Just before we start, I wondered if I could find out what stage many of you are at. How many of you are actually medical students at the moment? Okay, and how many are doctors in training? A few. Okay, that's good. So I'm going to tell you a bit about how my random path developed as a clinician-scientist. And what I discovered really doing this was 4 things that I think you might take away. One is that you need to find your theme really quite early and make sure that you get excellent training, not just as a clinician, but also as a researcher. But it's vital that you're a credible, extremely well-trained clinician as well. As Ingrid said, what you want to do is be prepared to grab opportunities and mentors as they come along. And what I'm going to go on at some length about is where my career ended up, which was helping other clinician researchers, especially young people. And finally, it's very important that you learn to collaborate. I think we've moved on from the model of the independent, focused researcher toiling away in their own laboratory or library. And we now are much more wedded to the concept of team science. So my interest in research really started at school. And this is my rat test from a month ago when I got back from Scotland, feeling a bit peaky. And I realised that it used the same very simple lateral flow chromatography methodology that got me interested in research in the first place while I was still at school. Because we had a new biology curriculum at school, which was largely experimental. And to pass the exam, you had to do a research project and write a mini thesis. And I was very interested in why leaves changed colour in autumn and what happened. So I did some chromatography, and I discovered that what happened early in autumn was that the chlorophyll, the green bit, disappeared, and there were yellow and orange pigments that got exposed. And then as autumn progressed, there were new pigments appeared that were deep red. And I wrote my little thesis on that. But in reading, I also discovered this fascinating phenomenon whereby the leaf actually falls from the tree. And that is that because there's a wave of programmed cell death at the base of the leaf that causes it to abscess, to drop off. And I didn't think any more about this for many, many years, but you'll see why that was relevant. Now, I was lucky enough to go to Oxford for my first bit of my medical training. And the great thing there was, although there were 9 terms, 3 years, only 5 of them were doing boring things like anatomy, physiology, and biochemistry, and passing the professional exam. 4 of them were really very much research-based. And I was very lucky to spend much of my research time here at the Savill Dunn School of Pathology in the MRC Cellular Immunology Unit, where together with another medical student who also became a professor, he's still in Oxford, I worked with a chap called Simon Hunt, who I discovered preparing for this talk had actually published our data without telling us. And that was what used to happen in those days. If you were students and you did things for a boss, the boss was able to publish your data. Because we did some work on looking for a marker called Thy1 on thymocytes in rats. And we were allowed to use this thing called a fluorescence-activated cell sorter that cost 5 houses. You'd have to sell 5 houses to buy this thing at that time. And that confirmed our hypothesis that there were indeed, this marker was on lymphoid cells. And this kind of got me interested in immunology and inflammation. Now, because I was a Londoner, I didn't want to go back to London to do my clinical studies. You could stay in Oxford, but it was suspect. And I thought I wanted to go somewhere really traditional, so I went to Sheffield. And this is the Royal Hallamshire Hospital at Sheffield, the main teaching hospital. And really I got fascinated by pathology. The best teaching was in pathology, and I realised that the basis of medicine was pathology. And at that time it was cellular pathology, histopathology, and so on. And here's my path, really, for the first part of my career. School, Oxford, Sheffield, that should say MBCHB. I managed to get the pathology prize, which was quite relevant. I was a junior doctor in Sheffield and Nottingham. And then I got my big break in academic medicine was I got a job that took me to the Hammersmith Hospital, and I'll show you a picture of that in a minute. After which I stayed there to be an MRC clinical training fellow. I'll explain what that is in a minute. Then I became a senior registrar in renal and general medicine again, and then I became an honorary consultant when I won a Wellcome senior fellowship, 2 competitive fellowships in that first phase. And really what I was doing during this period was I was living in this triangle that I hope all of you will end up living in, which is the clinical academic triangle, where service, research and teaching come together to boost the population's health and wealth. Now, this is the Royal Postgraduate Medical School at Hammersmith Hospital in London, a picture taken, I think, from the top of a bus. And this was a fantastic place to be a junior doctor because it was a meritocracy. There was almost no hierarchy. The top person you could call by their first name. And although I became a nephrologist, I'm not going to show you any pictures of kidneys at all, because the serendipity for me was meeting a chest physician who was interested in the same problem as me. Now, at that time, in the 1980s, the commonest cause of chronic renal failure was nephritis. And it puzzled me why some patients got severe post-streptococcal glomerulonephritis and yet that resolved completely, while others progressed to scarring. And I was lucky enough to meet a chest physician who was equally interested in this problem. Here's pneumococcal pneumonia. Here's a biopsy of the inflamed bit, and you can see the airways are absolutely stuffed with inflammatory cells, neutrophils. And my friend, mentor, PhD supervisor, Chris Haslett, was fascinated by how pneumonia could resolve completely, even in the days before antibiotics, with all those billions of neutrophils disappearing. And the dogma at the time was that these cells just fell apart, which made no sense at all because they're loaded with all kinds of dangerous proteins designed to kill bacteria. And Chris had gone to Denver on an MRC travelling fellowship. He developed techniques to isolate neutrophils and culture them without activating them so that he could label them, put them back into rabbits with pneumonia. And he showed unequivocally that the cells met their fate at the inflamed site. They didn't leave, they died at the inflamed site. And then when he cultured the neutrophils, he discovered this, that they actually changed morphology. And because he'd been a medical student in Edinburgh, taught by Andrew Wiley, who in 1972 had published a paper with Kerr, Wiley and Curry on programmed cell death, he recognized these as the changes of apoptosis. So neutrophils with these large multilobular nuclei, the nuclei condensed. And all these cells are neutrophils, and some of them look very different. So Kerr, Wiley and Curry had identified this programmed form of cell death, apoptosis, as opposed to what was then thought to be the only other mode of cell death, accidental cell death, murder, necrosis. And what they realized was there was a fundamental difference between these two means of getting rid of cells. Necrosis led to leakage of contents, inflammation and damage. Apoptosis led to non-phlogistic removal of these cells by the big eaters of the inflammatory system, the macrophages. And here is joint fluid from a patient with seronegative arthritis. This was, I was a training fellow. I went down to Mark Walport's rheumatology clinic and took some fluid from a patient with a knee effusion and realised that here were neutrophils undergoing apoptosis and being eaten and degraded by macrophages. And these appearances have been recognised 100 years before, and they were called Reiter's cells. And I spent the next 10 or 15 years of my career dissecting the molecular mechanisms by which these cells get recognised as senescent self and removed. And much of that still has to find any clinical application. But what we were able to do also was find ways of inducing apoptosis in inflammatory cells and their safe removal. And we discovered that eosinophils, if treated with steroids, die very rapidly by apoptosis, and the same steroids promote their phagocytic clearance, and that's probably why eosinophilic diseases are so steroid sensitive. And then working with Adriano Rossi, a pharmacologist, we discovered that drugs developed for cancer, CDK inhibitors, actually stimulated neutrophil apoptosis. And in a mouse model of inflammatory lung scarring, where you put bleomycin into the lung, once an inflammatory response has started, the mouse is destined to die of lung scarring. And yet what we were able to do was actually trigger apoptosis and prevent scarring with this drug. So what we see here then is the story of this piece of work. So Chris's original observation was 1985. Our paper was 1989. We learnt how to regulate apoptosis. We developed an animal model. We drove resolution by driving apoptosis. And then we got money from the MRC to do an experimental medicine study, and we chose the wrong disease. We chose a disease with immense unmet need, idiopathic pulmonary fibrosis. But what we should have done was choose a disease with lots of neutrophils to see if we could get rid of them. And unfortunately, that study had to be stopped because the drug was too toxic for patients with pulmonary fibrosis. So here's the lesson. This is a hero of mine, Sir Alistair Cook, one-time English cricket captain. He holds the record for the longest overseas innings at the MCG, 244 not out. He was on the field every minute of 5 days. And this man knows that cricket is a long game, and I think clinical research is a long game. And you can see 20 years, and we got nowhere with harnessing apoptosis. Now, what happened after that? Well, I became a clinical academic. I went to Nottingham to set up a new unit. I then went to Edinburgh to set up an MRC Centre for Inflammation Research. By accident, I became the dean, head of the college. I did all sorts of peer review work for chair of research boards. And then I worked part-time leading funders like the Scottish Government Chief Scientist Office and the Medical Research Council. And all this was because I got interested in helping other people, because I thought that I was going to have much more impact for patients if I could help others do research than just rely on my own research. And very soon after arriving at Edinburgh, I got a call from Mark Walport, who was the number 2 at the new Academy of Medical Sciences in Britain. And I got asked to lead a report on how to train clinician-scientists, and this was it. And it recommended the first thing we needed to do was count them, because no one knew how many there were, but we were convinced that they were decreasing in number. And Australia is at exactly the same point. We just don't know how many clinician scientists there are, but we worry that there aren't enough. And when we counted them, we discovered that there were almost as many as there were pilots in the Battle of Britain. around about 3,000. And what was very worrying was that the young pilots were dying at a fast rate, and it looked like the profession would be extinct by about 2008 unless we did something. And this gave us great impetus for trying to influence government and funders and others to put together a program to really train clinician scientists in a way that enabled them to become a good clinician and become a researcher. And we grabbed the opportunity of a new training program in Britain called Modernizing medical careers. That meant that in 2006, Mark Warport was able to convene a committee. I was on it. A number of other people were. And we developed this program. I won't go through it, that integrated research into postgraduate clinical training of doctors. And it's been very successful, sustained in Britain now for 15, 16 years. And you'll notice that the PhD element of this is done while people are registrars. Now, you can do a PhD at medical school, that's great. The problem is that your PhD is ancient history by the time you've become a consultant and able to become an independent researcher. Or you could do a PhD at the end of your postgraduate training. That's fine, but you have to accept 3 years of almost no wages. You've got a family, a mortgage, all this stuff. And what we were able to do was point to data from the Medical Research Council that showed that people who did their training fellowship like me when they were registrars had a fantastic career. So basically, do it that way, and two-thirds of people remained in the academic profession, which was a really very good return on investing in registrars who wanted to do research. When I got to Edinburgh, we started to develop our own scheme, the Edinburgh Clinical Academic Track. And then when I came to Melbourne, I discovered that at least in my part of Melbourne, there wasn't any systematic approach to an integrated training program. So we've set up the MAC Track, which is now in its 3rd year, where we integrate PhD, pre-PhD, PhD and post-PhD research training into registrar training. And this, if you like, is a pilot for a program that the Group of 8 is the 8 big universities in Australia, includes Monash and Melbourne and universities like that. And a guy called Stuart Carney, who's the Deputy Dean at Brisbane, who was a junior doctor representative on that Warport report in Britain, led a committee that came up with this proposal to the Australian Chief Medical Officer's workforce inquiry. So there is a proposed track, and I think over time this will get implemented. It will require some extra money, but perhaps not as much as we feared. Now, I'm going to finish by talking about learning to collaborate. And you start doing that because the guy or girl next to you on the bench is doing something interesting, and the two of you talk over coffee and you start collaborating. But what's really, really important now in clinical research is collaborating between disciplines. This is the Queen's Medical Research Institute at Edinburgh. Chris Hazlitt and I raised $100 million to build it in 2 years because Chris had this vision of interdisciplinary collaboration, inflammation, reproduction, cardiovascular, all come together with shared research problems and themes. For example, all the physiological processes of female reproduction are inflammation. Ovulation, implantation, parturition, they're all inflammatory processes. So here was a great example of collaborating between medical disciplines. And then certainly in Britain, this is Patrick Maxwell, who's the Regius Professor of Medical Science in Cambridge. wrote a very influential report on engineering and physical sciences in health and life sciences. Now here's the new MRC Laboratory of Molecular Biology. That's the UK's Nobel Prize factory. If you get a chance to go there, go there. Fantastic place. The Queen opened the first one in 1962. You can see things were a little bit formal then. And they were less formal when she opened the new one in 2013. And I took her as the chief executive of the MRC to meet 4 PhD students. They were a mathematician, a computer scientist, a physicist and a chemist. And they were all in this medical biology institute doing medical biology. So these institute things are important. Am I over time? Oh, right, okay. And really, really important is this set of inter-institutional collaborations in Australia between research organisations, healthcare systems and industry. Helena Teed leads Monash Partners. She might tell you about that. I'm involved with the Melbourne Academic Centre for Health, which brings together 10 health services, 8 medical research institutes and a big university. It serves over 2 million people, enormous collaboration that I think will have tremendous impact on research and translation. Now, I want to just digress for a second or two about something that really is important in this focus of collaboration, and that is linkage of electronic medical records. I did my PhD more or less entirely using that, and now I suspect that you're going to use this when you do your PhD. Much of what you're going to do is going to be in silico, it's going to be data linkage and so on. And I got turned on to this in Scotland, where there's a fantastic system for collecting every day from every source in the country all the clinical data on anyone with diabetes. And just collecting that data informs clinicians about the quality of care and has resulted in dramatic improvements in outcomes. For example, a big reduction in amputations in part of Scotland called Tayside. And it's opened up new ways of doing clinical trials. So the very first trial that showed that statins were beneficial in coronary artery disease was done in the west of Scotland, where many men of 40 dropped dead with heart disease. This is Ian Ford, the biostatistician. And what Ian did was he followed those patients 15 years later by electronic records linkage. And he showed that those who'd been randomized to pravastatin still had an enduring benefit. And he published this in the New England Journal of Medicine. Now, the original trial had cost £30 million. This study cost £30,000 and demonstrated a benefit. And it's led in Scotland to a change in the way that clinical trials are done. They're now done on real-world data. So you simply find patients in the system who are having the intervention you're interested in, and you compare them by records linkage with controls, and you can define whether or not that intervention is beneficial. So it's a very powerful way of doing trials. One of the things I was involved in at the Medical Research Council was setting up Health Data Research UK. And what this shows is the power of institutions collaborating, because it's a distributed institute across the whole of the UK with sites in all the UK countries. While I was at the MRC, we did the same thing with dementia research, because you get much, I think, more impactful research if you can get groups of people across a country working together on a problem. And we extended this to be a platform with industry, so that we could work with industry on trying to find new dementia treatments. And we had all sorts of other collaborations with industry. It's interesting that Weehai has a fantastic centre for drug discovery, really lead discovery. The way we did that in Britain was that we just teamed up with AstraZeneca and we used the industry facilities. So to finish, find your theme. I got interested in research at school and I got interested in inflammation as a medical student and a young doctor. Be prepared to grab opportunities and mentors. I happened to bump into someone interested in the same thing as me, who became my friend, mentor, PhD supervisor. Help other clinician researchers. Well, I've spent an awful lot of time on trying to find pathways for people like you so that you can do research and clinical medicine at the same time as you train up, and crucially learn to collaborate. Now, I just want to finish. These are 4 people I owe an awful lot to. I've mentioned Chris. Keith Peters is a lifelong friend. He was the man who appointed me to the renal unit at Hammersmith. Colin Bird was the professor of pathology in Edinburgh, who persuaded me to move to Edinburgh. And David Carter was the chief medical officer in Scotland when I got there and got very interested in training young doctors. I was lucky enough to be funded for many years by the MRC and the Wellcome Trust. And I'll just leave you with that, because I hope you have a lifetime in that triangle and that you enjoy yourselves as much as I have. So I'll stop there. Sorry if I've gone on a bit.
So thank you, Sir John. That was fantastic, and I think it gave you a glimpse of the science and the career and the positives and the negatives, or the challenges. It wasn't really very negative. It was great. Have we got our microphone holders? Can one of you go down this end and one down that end? And please pop your hand up if you have a question. Now, we already have a few online, so thank you very much to the online people. So the first one online is, if there are no what would it take to see the Clinician-Scientist pathway taken up in Australia? And is there anything med students and others can do to help to bring that about?
Well, I think medical students can help enormously by getting active in research while still at university. And again, lots of Australian courses give you that opportunity, and you'll discover that the medical student research societies are often plugged into what the junior doctors and the senior doctors are doing. The Academy is doing great things in this area, and I think that these pathways will evolve quite rapidly in Australia because of the leadership from the Academy and the big universities.
Yeah, I mean, the Academy is very keen on trying to get a much more defined pathway in the system, as you do in the UK. And I had one of the Foundation One just post-med school interns come out and work with me, and for 6 weeks or something. She was incredible. So you're obviously getting the right people in those training pathways. Do we have any questions in the room? Yes, please. Wait a sec, the mic is on its way to you for all the people online.
So you mentioned that you had this long-term research which ended up stopping because of the toxicity. How do you cope with that long-term, I guess, knowing that it doesn't translate clinically? Like, is there a way that you go about moving on, or does it affect you at all in the first place?
Right, so I got very interested in discovery. So I think that there are some important discoveries that were made that have been corroborated and repeated by others that one day will have clinical application. It's just not yet. But it's interesting, you know, this field has grown like topsy in terms of clinical application. My story of woe is almost exactly the same timing as BCL2 to venetoclax for blood cancers at Weehai. Fantastic success. And any of you know about the treatment of cancer will know about checkpoint inhibitors they hit this pathway of programmed cell death 1, which is a receptor on immune cells which actually stops them dying. So this area has enormous clinical application. It's just that my little bits of it were enough to get me into the Royal Society, but they haven't earned me any money yet. I'll pay back, and might never do.
We don't do it for the money, do we?
No.
But the Royal Society is the most elite group of scientists in the world, so this is not a small thing, this is a big thing. There's a question just there. We'll take that and then I'll do an online question.
I will ask a question about money because I thought it was really interesting how you created the consortium with industry in mind, and understanding that currently we're in this age of discovery but also translation, where we have emerging biologic therapies where the first dose of Beam 101 was administered in New Zealand. Clearly industry and all these innovators in pharmaceuticals, therapeutics development are identifying Australia and New Zealand as the key target locations for clinical trials. How do we empower our clinician scientists to better equip themselves with the skills to engage with industry, because clearly they're interested in the expertise, the patient population, the level of engagement and the research potential. So how do you see Australia or your role in bridging that gap?
So I think an important thing is that when I was your age, people were very suspicious, academics were very suspicious of industry, because they thought that all industry wanted to do was pursue its own agenda and tell them what to do. And what we've discovered over the last 20 years is that our colleagues in industry are exactly the same as us. What they want to do is discover and translate and implement more effective treatments, diagnostics, devices, and so forth. So actually we start in the same place. So I think the thing that we can do is attract industry to us, make it easy for industry to work with us. And one of the things I'd like to see developed in Australia is a system similar to that we've had in Britain for 15 years, biomedical research translation centres, where investigators like us can try out new treatments with the help of patient volunteers, and we can attract industry to partner us and do those things. Because what industry brings is enormous resource knowledge that we can use to make discoveries and improve treatment. So don't push them away, pull them towards you.
But pull them towards you with integrity.
Absolutely.
And I think that's really important. I do a lot of pharmaceutical trials, probably about 15 at the moment, and they're driven by pharma, which is completely different to what you're talking about, which is where it's driven by the clinician-scientist. And so you have to have a relationship with industry, a very good one, but you have to remember which bit of that which part of that playground you're playing in, and you have to manage it. Would you agree with that? Yeah. Okay, so another question which sort of relates a bit to the previous one, but I just want to give you another opportunity around the topic. If there are no formal pathways in Australia for junior doctors to become clinician scientists yet, what should we do to gain the skills and experience needed?
Well, I think to be a clinician researcher, it's essential that you do a PhD. And there are many ways of doing that. These integrated pathways are only one way of doing it. You can do it at medical school, you can take time out as a registrar, you can do it as a new consultant. That for me is the sine qua non, is get into a really good research group and start training with a PhD. And most modern medical curricula enable you to find your theme by doing some small research projects or whatever with supervisors in your medical school. So I think that's the thing, is get a PhD.
I agree completely, but I think, there's a question down here if the mic could come. But I actually loved your comment, John, about a PhD in med school being ancient history. I thought that was spot on. And one of the big issues, I think, in the US is they do an MD PhD program and it's always basic science. And I'm a passionate clinical researcher, and you can only do clinical research once you see patients and you know how to assess them. So you actually can't do clinical research until you're a clinician. And that's why I loved what you said about a PhD being later. Please.
My question was about your first comment on finding your theme. As a medical student, there are lots of areas that are exciting, and I think that there's value to be added. So how do you pick which theme that is to, spend the rest of your career focusing on?
Well, it's often an accident. So at medical school, I was equally interested in neuroscience because my grandfather had died with multiple sclerosis. And, it's just that the inflammation side lucked out. And people often find their theme as a junior doctor. They see an interesting patient. I got the job at the Hammersmith by claiming I'd been turned on to research in kidney disease because I saw a very interesting patient who, whenever they got staphylococcus in their nose, they got proteinuria from their MCGN. So it can happen at any time. But what I mean is I think the thing you need to do is find a thing and stick with it. Because generally we start later than basic scientists. We haven't got time to do butterfly stuff. And what we're interested in is going to take a career to translate. That theme will come at some stage. It might only come when you get your PhD.
So you can change tracks, but building is really exciting because you become a world expert in your area. We have another question online. Thank you very much to the people online. Great questions. How do you find opportunities to visit other researchers and institutes?
Well, again, that's remarkably easy. I think for medical students, setting up a research society, inviting local researchers to come in and meet you and speak is definitely a great thing to do because, you saw all these wonderful institutes. There's a whole lot more in Melbourne. You could get researchers from them to come and meet you, talk with you. Researchers are always delighted to speak to young people because they may turn up as your next postdoc, some years in advance. So I think that's the thing to do is get involved with researchers.
Great. Any more questions in the room? Well, if not, thank you very much, John. And here's a little token for thanking you for giving up your weekend and preparing that lovely talk. And thanks very much for joining us.
And I must apologise to everyone and Christabel for having to go and fix my son's door.
Thank you very much. Great. Thank you. Oops. Thanks, John. All right, so I'd like to move on to our next keynote address, please, and it gives me great pleasure to introduce Professor Christabel Saunders, who is now the James Stewart Chair of Surgery and the Head of Department of Surgery at the University of Melbourne, a consultant surgeon in general surgery at the Royal Melbourne Hospital and the Peter MacCallum Cancer Institute. And I think we've been very fortunate to just draw her away from WA, where she was. So a great gain for Melbourne. Christabel is internationally recognised as one of Australia's most prominent research-oriented cancer surgeons and has substantially contributed to breast cancer research, including trials of new treatments, psychosocial, translational and health services research. She became a Fellow of the Royal College of Surgeons in the UK in 1991 and in 2003 became a Fellow of the Royal Australasian College of Surgeons. She became a Fellow of our Academy in 2016 and received an honour of the Order of Australia in 2018. So Christabel, we're really interested to hear your journey.